Hello, my name is Lana Vasung, and I will give a talk titled The Unique Challenges and Importance of Incorporating Prenatal Brain MRI into Lifespan Models. I do have to mention that this talk has been adjusted for the audience that is less familiar with the field of fetal brain development. And in addition, I will focus mostly on the unique challenges, and I do hope that by acknowledging those challenges, you will understand the importance of incorporating prenatal brain MRI into lifespan models. I will start with a small introduction. As you know, recently there has been an increase in the amount of research aimed to predict the brain age across the span. And one idea behind this effort is an attempt to detect early a potential disease. And the idea itself, which is characterizing age, is an old idea that sparked a lot of discussion in the field of medicine, especially fields of neuroanatomy, embryology, and gynecology. So in the field of gynecology, fetal age is determined by the postmenstrual age of the mother. So although this approach is convenient, postmenstrual age is not an age, and it often comes with an error spanning from approximately several days to weeks. So to increase precision, embryologists and neuroanatomists use post-fertilization, post-ovulatory, or post-conceptual age, which I will use in this talk. And the most accurate assessment of the fetal, or that is embryo age, is the measurement of the crown rump length, or the greatest length. And compared to the postnatal lifespan, this early in pregnancy, except for the watchful waiting, which is the source of considerable stress for parents and physicians, very little can be done if there is an offset or an error in determining age. Moreover, acknowledging that something is different from what we expect at a given age very often comes with the question how and why this is different. And this is something that I will try to address in this talk about the prenatal fetal brain development. You will notice 5 take-home messages from this talk, and I will start with the first one, which is public access web-based portals for visualizing, searching, and sophisticated mining of fetal structural and functional neuroimaging data are virtually non-existing. And this message is kind of linked a little bit with the work from Richard and Jacob that was recently published in Nature, titled Brain Charts for the Human Lifespan. So they aggregated possibly the largest MRI data set ever. And for postnatal lifespan development and aging, that is periods of neonatal brain development, child brain development, adolescence, or aging, they could easily turn to open access large cohorts, such as Developing Human Connectome Project, Enigma Imaging. However, if one wanted to analyze and the prenatal data, which showed to be highly relevant, as one of the studies pointed out here on the left, is that the velocity of cortical thickness peaks before birth. So if one wanted to include the prenatal data, one should turn to individual researchers with the relatively small data set of fetal MRIs that were acquired and processed using in-house protocols. So the knowledge about the prenatal brain development lies in the small number of dedicated researchers, which brings me to a second message. We lack harmonized multimodal cross-site MRI acquisition protocols, and on top of that, we're limited with a number of MRI sequences that can capture fetal brain structures that are often transient and state-specific. So to elaborate on this, as you probably know from neuroimaging and neuroradiology, there are a number of MRI sequences that one can use to better characterize the brain structure or function after birth, as you can see in the top row. However, in comparison to postnatal lifespan imaging, there is only a handful of sequences we use nowadays to reconstruct and quantify fetal brains, as you can see in the bottom row here, haze, trophy, and there's some white. Which brings me to my third message linked with the previous one. There is incompatibility of fetal MRI processing pipelines that is linked with challenges of the fetal MRI segmentation and challenges in cortical pixelation. So the main problem here is that we're currently limited in ways we can connect prenatal and lifespan findings. So to elaborate on this, the first thing to mention is definitions. So during the postnatal lifespan, the cerebral cortex is defined as an outer layer of neuronal tissue that contains neurons and functions in coordinating of sensory and motor information. However, you will notice that during the prenatal development, the fetal brain, as you can see in the image on the left, in the left upper corner, the fetal brain has several outer layers, not just one. And that the outermost two layers, the transient fetal zones called cortical plate, CP, and subplate, SP, contain neurons, as you can see in the NISO on the left. So if we go one step further, the cortical plate is an outer layer that after 20 post-conceptual weeks contain 90% of post-migratory neurons that are assembled in 6 cortical layers and is often referred as to future cortex. However, the first synapses that transmit and possibly coordinate sensory and motor information are actually found in the layer below, that is the transient subplate zone. So on the right, you can see the number of synapses throughout the different cortical layers, different fetal layers. So definition of fetal brain layers and their gradual disappearance poses significant challenge for fetal brain segmentation. And speaking about the cortical plate and the subplate, you can see if we measure the thickness of the cortical plate, that is future cortex, it kind of reaches the 2 millimeters very early, around 20 weeks. And it remains more or less similar throughout the entire mantle up to newborn periods. In contrast to the cortical plate, you can see the subplate zone changes dramatically, shows large spatio-temporal variation in the thickness, and then gradually it disappears after birth, which sometimes we cannot characterize by the magnetic resonance imaging. So again, the definition of fetal brain layers and their gradual disappearance poses a significant challenge for fetal brain segmentation. As you can appreciate by the figures here, and these are only for visualization purposes and they're not to be used for quantification, from post-conceptual weeks here up to 40 gestational post-conceptual weeks, there's a tremendous variation in spatio-temporal signal intensity of transient fetal zones. So one needs to develop and tailor segmentation algorithms for particular gestational age. That is, until we have large longitudinal fetal MRI datasets. And as you can see in the row below, up to 32 gestational weeks, we're successfully segmenting all the layers, almost all the layers in the fetal MRI. But after 32 gestational weeks, we lack tools, in particular because we rely only on one haste sequence. I have to mention that there has been a tremendous amount of effort from FETA team to organize fetal MRI segmentation challenge that would be open to community as a part of MAKI, which I'm advertising here, which moves me to another associated challenge, parcellation of the fetal cortex. So let's put on the side debate, what is cerebral cortex in the fetus? That is, will you combine the cortical plate and subplate and call them together fetal cortex, or you will just look at the cortical plate? So for simplicity, let's say one wants to look at the cortical plate only, like I did in the image, top left image. So if one wants to parcellate the cortical plate only, one can use sulcal landmarks. And we know from all literature that cell size start to appear early, around 18 post-conceptual weeks. And as you know, there has been a long-standing debate whether these sulcal landmarks represent borders between functional or architectonic areas, as the left image below. And I'm not going to go into that debate. What I want to show you here is that whatever you think about the postnatal and adult brain, it doesn't really apply to the fetal brain. As you can see in the right column below, the adult cortex can be parcellated into different cytoarchitectonic areas based on the cortical layers. However, some of those areas do not exist in the fetal brain, as some regions will lose cortical layers from the 6 layers, and some will gain them, as you can see in the bottom row of the upper right image. So this brings me to the next challenge, where one seeks to identify what drives the gain or loss of specific cortical layers in specific regions. And here I have to say we don't know, as we lack comprehensive fetal gene expression maps and histology immunohistochemistry maps. I will give you an example. Recently, in our publication, we tried to link gene expression with changes in thickness of the cortical plate and subplate. However, aside from 21 to 22 post-conceptual week fetal data, we could not find cortical gene expression data that covers period from 32 post-conceptual weeks to newborn. That is periods during which the cerebral cortex undergoes the most dramatic changes, from gyrification to ingrowth of thalamocortical fibers and resolution of the subplate. And in my final take-home message, I want to touch on the topic of fetal brain reorganization and plasticity. That is, what is the meaning of the structural deviation before birth and how sensitive and specific certain findings are. To give you an example, during prenatal brain development on the top left, the corticospinal tract that carries axons for movement-related information from cerebral cortex to the spinal cord grows. So the corticospinal tract reaches cervical levels, which are important for the movement of the upper extremities, around 15 post-conceptual weeks, and continues to grow towards lumbar levels, reaching them around 27 post-conceptual weeks. However, the amount of pyramidal tract that decusates by newborn age varies significantly between individuals. So does the thickness of the subplate zone, especially in these regions M1C and subplate zone that is often called axonal waiting compartment. You can see on the right in the blue dots the thickness of the subplate zone from 20 to 30 gestational weeks. And subcortical injury during the prenatal period, and the bottom right, when the corticospinal tract is growing and the subplate and the cortical plate is under significant reorganization, can leave structural consequences such as volume loss and loss of ipsilateral cortical spinal tract without resulting with minimal functional behavior consequences. Why? Because the fetus is moving long before the cortical spinal tract reaches the spine, which probably plays a significant role in retraction and reorganization of the corticospinal fibers. Finally, certain conditions such as isolated ventriculomegaly are characterized by overgrowth of the proliferative zones at the future cortex, like you can see on the left, and slower growth of the subplate, but are generally associated with normal postnatal development. In contrast, brain development in congenital heart disease on the right is also characterized by slower growth of the subplate zone, requiring a better definition of histologic, genetic, and neuroanatomic substrate underlying these quantitative changes. Thank you so much.